Afternoon. Myself, Rishikesh Kulkarni. I am an assistant professor in the Department of Electronics and Electrical Engineering. And this work is with my MTech student. This is one of the first work of laser speckle based contrast imaging for agricultural applications, which we have done. He was my MTech student, along with another professor, Professor Pranab Mondal from Mechanical Engineering Department. So I'm going to talk about the application of laser speckle contrast imaging for plant and seed characterization. So let me briefly explain what is speckle. Here, speckle is a granular random intensity pattern. When you eliminate any optically rough surface with a coherent light source, such as such as laser light. Now, if you eliminate any object with white light, you not observe this kind of pattern, which is shown in this figure 3. However, because of the coherence nature of laser, you observe this pattern because it generates a very complex interference generated by the coherent light source upon the reflection from optically rough surface. So in speckle contrast imaging, as you can see, this image is a totally random image. So this can be recorded with two optical configurations where you can see that it is illuminating the optically rough surface, and I keep the CCD or CMOS sensor in front of it. When I record such a light, we call it as a objective speckle pattern, whereas if I keep an imaging lens and use the imaging lens to record the light reflected from the object, that type of speckle is known as subjective speckle. So one of the example which I have shown here is the subjective speckle. As you can clearly see, it is a totally random image, does not carry any information as such. However, if you record such speckle pattern over a period of time, then it does contain the information of the state of the object. So that is where this laser speckle contrast imaging is useful. It is very widely used in bioimaging. Many commercial applications are also already available. So we are actually entering into the application in the agriculture domain. Now here I'm showing a simple video where we have recorded this plant, the leaf, part of the leaf, which was illuminated with a laser light source. And when you record a video, you can see that in this portion, you can see some activity which is going on, where we call it as speckle boiling, more activity known as speckle boiling, whereas this other laminar portion, there is no such activity. Now, this is a small video. If we analyze this video, we will be able to identify which location of the leaf is more active, which is less active. And we have used this to find out if you subject the plant to different conditions, like biotic, abiotic conditions, let's say low pH, high pH, low water supply, or if the water supply has salinity in it. So all this can certainly be, to certain extent, quantified with this method. Now, the application of this has been reported in many literature available, such as it has been used for understanding the fruit freshness. So if the fruit is fresh or not, if you can analyze it without cutting the object, like apple or mango or orange, that was reported in the literature, plus some non-agriculture applications such as drying of alcohol on a surface or something like that. This also has been explored with this technique. Now, the focus of this work was also develop the speckle imaging technique setup, plus developing an algorithm that how do you analyze such speckle images which are recorded over time. So these two, that is Fuji algorithm and generalized difference algorithm, are the standard techniques which are used to analyze the speckle images. Here I'm representing the intensity IN as the intensity which is recorded at nth time instance. So here what we do is we compare the intensity recorded at nth time and n-1th time. So if you take the difference, if there is any activity, then this difference will be non-zero. If there is no activity, this difference will be 0. So this is the usual approach which is utilized in the Fuji algorithm. Now what we have proposed is we have utilized a weighing factor, Wn, as you can see in this equation, which provides a weight to the quantity which is shown in the modulus here, so that the images which are having more activity are considered in the calculation of the contrast in comparison to the images which are not having that much correlation. So with this weighing factor is calculated using the correlation algorithm between two intensity images, and this weighing factor is going to produce a contrast which we expect to be better than the Fuji algorithm. Now, this same approach can also be considered with the generalized difference approach, where, as you can see, the difference of two consecutive intensity patterns are considered here. But in this, you take the difference of all the possible combinations of intensities. So the algorithm goes like this, that we record set of images and we take the difference between two consecutive images, multiply with the weighting factor, which is nothing but 1 minus the correlation of the two intensities, and then we compute the quantity FTC, which we call it as the contrast map, and then we smooth it with a median 3x3 median filter, and we go on doing that at each instant of time n, and we combine it together. Now, to show the effectiveness of this approach, so what I've done is that this is a image which shows different regions, black to red, and as you can see that these different patches show different activity, and the activity shown here. So the black one has a flat part on the y-axis. We can see that means there is no activity in this region, whereas there is a highest activity in the middle, because there is a huge decorrelation between intensity patterns, and the correlation varies in between regions. So from R1 to R5, the activity is increasing. So if you record a speckle image for such an object, you can see it is a random image. We cannot make out anything. But if you record the intensity over time, because of the object is getting decorrelated, you can see different regions here. Now, this is the result obtained with Fuji. This is the result obtained with GD method, which are the standard methods. But our method, as you can see, has clearly given the contrast. Like we can clearly identify which region is more active, which region is less active. So this, from the contrast perspective, if you look at qualitatively in the image, we have obtained the better results. Quantitatively also, we calculated in each region the noise factor, which we define it as the standard deviation divided by the mean value within each region. And in each section, we have found that our method has less noise, that is better signal to noise ratio. Now, experimentally, as you can see, it consists of very simple component. You need a laser source. You illuminate the object with the laser beam, which is expanded by the lens, and you have a camera to record the image. It is a normal industry grade camera. The specifications are mentioned here. Now, for the leaf example, which I have shown before, this is a speckle image. Again, you do not make out anything from here, but when you analyze over time, you can see that you get a contrast. This contrast map for Fuji and GD are shown here, which do not very clearly identify the region of higher and lower activity, but you can see here that the red region is the vein through which more activity is happening in the leaf, which is coming out to be red in color, and the yellowish and green color show lesser activity in comparison to the green region. Even there are some spots here which are yellow within the range region, which were identified only by the proposed algorithm in comparison to Fuji and GD. So you can clearly see that by quantifying this contrast map appropriately, you can actually find out the effect of different biotic abiotic stresses which the plant is subjected to. The same experimental setup was used to study the seed viability. What is seed viability? We'd like to know whether a given seed sample is able to germinate or not without actually germinating it. So here, the most common test is the TTC test, tetraphenyl based test, wherein you basically subject this seed to a temperature of 35 degrees for 40 minutes, and it is kept in a tetrazolium solution. Then you take it out. Then, depending on the color of these seeds, you identify whether the seed will germinate or not. You can see this beet red color shows that the seed is good to germinate, whereas when it is not colored, we call it as it is not suitable for germination. That means the seed is not useful. Now, we wanted to know whether this technique can be utilized without subjecting it to TC test, which is a chemical-based test. What we did is we used the same setup to eliminate the seed with laser light. We found out the contrast map, and as you can clearly see here that higher activity indicated with red region directly corresponds to the seed which is of higher viability compared to the seed which is having low viability. So we actually had 3 samples In fact, what we did was that we subjected the seed to different temperatures to make them non-viable. So when we made it and we compared with the conventional TTC test, we found that we can certainly use this with direct correlation with the conventional TTC based algorithm. The same algorithm to verify whether the proposed method is accurate or not, we actually utilize the corn data set which is already available, made available by the authors in the paper which is mentioned here. And we use this algorithm in comparison with Fuji and GD. And here also we could see that the activities which are shown with the corn seed are also highlighted very well by the proposed Tinker algorithm compared to Fuji and GD. I've also mentioned the computation time taken by the proposed algorithm, which is slightly higher than Fuji algorithm, but significantly less than the GD algorithm. So the activity in the endosperm to embryo is clearly visible in this. So to conclude, we have developed a novel speckle image processing algorithm to analyze laser speckles, and comprehensive performance analysis assessment of the proposed algorithm was done with simulation as well as experiment in different conditions. And the validation of the experimental setup was done with leaf sample and seed viability testing. Thank you.